Good morning. I'm Brian Stephenson. I'm a professor of plant pathology at the University of Minnesota in St. Paul. And we're standing right now in a field in Crookston, Minnesota at the Northwest Research and Outreach Center. And specifically what we're doing in this plot is working with a disease called bacterial leaf streak of barley. And that is caused by a bacterium which we call Xanthomonas translucens pathovar translucens. And this is a pathogen that can really cause severe losses on barley. And the way that it's actually spread is that it can be seed-borne, so the bacterium can get in the seed, and so when you plant that seed the next season, it can be infected in the first part of the seedling phase of growth. So that's one way that the disease can spread from year to year. The other way that it can spread is actually through crop residue. So if you have a heavily infected barley crop and then you were to recrop on that same ground, the bacterium can survive in that residue and then infect the newly sown crop. And then the third possible way that we can see this bacterium be transmitted from seed to seed is sometimes on some of the perennial weed hosts that we might have in areas where cultivars of barley are planted in this region. So one of the main ways that we can control this disease is actually by developing resistant varieties. And so this is our main way of control because there are no fungicides that can control this. Fungicides do not work against this bacterium to a really strong effect. There are, of course, bacteriicides, but they are not practical or economical to control this disease. So I want to tell you a little bit more about this specific plot that we're in right now, and I'm going to call into the field of view my graduate student, Mitch Ritzinger, who's going to tell you about the screening process that he's doing in this particular plot. Mitch?
Yep. Hi, so I'm Mitch Ritzinger. I'm one of Brian's graduate students. I started about a year ago on this project, and essentially my goal is to find some sort of genetic resistance in any germplasm, in any barley variety that has been grown around the world, so that we can find it, document it, and then we can publish on it and ideally bring it into a breeding program, which we have here at the University of Minnesota, so that breeders can utilize this germplasm and the genes that we find and bring it into useful barley varieties that we already grow and we use locally. So in this nursery, you can see there are a lot of plants that are very down to the earth, very prone, and things that look nothing like what we grow around here. That's because Brian and I have assembled a very large, very diverse set of germplasm that's from all over the world. We have roughly 3,000 different accessions here that are from different parts of the world, whether they be seed banks in Germany or from the IPK in England, or we even have a set of germplasm from Eritrea and Ethiopia, which are both located in East Africa. So we've basically taken as much diversity as we can get our hands on and thrown it into a field here, and we have them planted side by side, and we fill up a backpack sprayer, which is kind of like a leaf blower with a water tank attached, and we take the bacteria off of a plate and put it into the backpack, and we walk through and spray them on all the plants so that they get an even coating of the bacteria, and we see which ones get the sickest and which ones get the least sick. And then we use a 1 to 9 scale, which is a rough percentage of a 0 to 100 scale, to approximate how sick each of the plants get. And then as we move forward in time, we can see how they progress and which ones eventually by the end of the season will completely die off due to this disease. And ideally at the end of the season, we'll still have some left standing that we can say, wow, these guys have withstood some tremendous pressure from this disease, and we'll be able to move those forward into the breeding program and document the resistance.